Good morning, everybody. I trust all of us, most of the participants are here. I hope you can hear me. I'd like to welcome everybody to this year's ARCS Meet and Greet event to introduce this year's cohort of 16 UCSC ARCS scholars and to celebrate them. My name is Almut Wolf, and I'm the Assistant Director of Foundation Relations. I'd like to start us off with a couple of housekeeping items. As a reminder, the meeting will be recorded. Closed caption is turned on, and you can select this feature on your Zoom screen. Please keep your mics muted, and ideally your cameras on. And I'd like to keep questions to the designated Q&A section at the end of the Scholars Talk. You can then informally ask questions or put them in the chat. In my role, I have the honor of being the liaison to the ARCS Foundation, and every year I assist with coordinating two signature events for our scholars, namely this online meet and greet and the in-person Scholar Symposium in April of next year. ARCS scholars, welcome and congratulations on being awarded an ARCS scholarship this year. You were nominated from your departments and selected from a pool of outstanding candidates. This is a financial recognition allowing you to decide what is needed to achieve academic success. But it is also a prestigious award recognizing your work and your achievements. ARCS guests, welcome to all ARCS members and ARCS executives. It is a distinct pleasure to welcome you, and I'd like to thank you on behalf of UC Santa Cruz for this partnership that spans decades. You have funded a stunning total of about 400 students at UC Santa Cruz, which we read as an expression of the trust you place in our programs and in our students. I would like to extend a special welcome to Heidi Hutchberger and Judith Webster, co-VPs of University Relations, who have organized today's meet and greet with me. And UCSC leadership, welcome to Dean Beal, Vice Provost and Dean of Graduate Studies, and Don Smith, Distinguished Professor and Associate Dean of Graduate Studies and Postdoctoral Affairs. They both work on the initiative Inclusive Excellence in Graduate Education to ensure our students and PhD programs remain among the strongest in the nation. Without further ado, I'd like to hand it over to Peter.
Yeah, thanks so much, Almut, for your nice introduction, for welcoming everybody. Again, from the bottom of my heart, this is really one of my highlights of the year. Actually, I would love to see you live, but I will see you live in April in San Francisco, the second highlight. And thank you, ARCS members here, board members. You know, most of you I have met over wonderful you know, dinners at the reception, and it's just like an amazing opportunity to be here, to see you all, to thank you all, and because we are so proud of you. So this is not about us, me and Don. We will give you just a quick presentation, about 10.5 minutes, I think we got, Almut, no, this is not about us, but nevertheless, and we share this presentation for you, especially for our board members here from the foundation, to see what we are doing, maybe new things, but some of you as graduate students, because I know that you're mostly in your lab and don't look at strategic initiatives, what the university is doing. So maybe for you also, just a little bit an update. So first slide. Yes, so I would, you know, maybe raise the hands. No, I'm not. I don't put you on the spot. This is our Leading the Change new booklet, new plan, strategic plan. And you graduate students have helped us to bring that together. I talk at the end a little bit about that because we're really breaking ground to something extraordinary happening at our university. Next one. This is our team getting bigger. Even our board members from ARCS, you know, you realize we're getting bigger. So we doubled actually, you know, since a couple of years, because we do need to do a better job in this environment where we really focus on one thing, and that is student success. So we have new members, and you can look through them. They're also on the website. And I'm really happy that we have more staff helping you to go successfully through your program. Next one. This is just like a little where we are right now. As you see, we have about 224 new doctoral students. So it's not just you, but there are more. And I really encourage you. I do that every year. Please also get out of your lab. Go into events of the Graduate Student Commons, to be engaged in the GSA, because it's about you, about your voice. And so we have 224 doctoral students and 351 master students. And look how wonderful we're coming together at the bottom here on the left, when you really see how diverse, how we are moving really in the right direction to make our campus, our graduate students look like the country. So we're very happy, especially since we are now an HSRU, Hispanic Research University, Hispanic Serving Research University, one of 21. Talk quickly about it at the end. So these are pretty good numbers. We are steady in regard to our international. We are steady in regard to our admission rate. So we are really, really proud of the diversity we are bringing to the campus. Next one, please. And just quickly, that you see that you are part of where we go, how elite you are, that you will, that you have been actually admitted, because as you can see, our admission rate is really, we're getting more and more difficult to get into, which is definitely in regard to our ranks, in regard to our AAU, American Association of University membership. So here is just this trend in regard to applications going up, and also in regard to the numbers, both for PhD and numbers here. Next one. Especially interesting for you in the AXE Foundation, as you can see, in the STEM disciplines, what we are focusing here on. We have, of course, here in engineering, a slight tip, more or less steady in the PB-Sci, physical and biological engineering. So this looks all good in regard to, if you see and what you know, you graduate students, what happened last year with the labor action and our support for you. So clearly, the salaries went up, and we are still able to support you in this way, as you can see here. So a very soft landing after that. Next one. And we really here overall, as you see, very healthy enrollment development, which is important for the ARCS Foundation to really see that we are stable and we're moving in the right direction, especially also in regard to the master students, which are going up, because we have also to think about the California workforce. Next one. And that's where we are right now. So we had a soft landing even after labor action, and increase up to 30% of salaries. So we were able actually to bring still in the best students, and you are some of them. So we have about 224 new doctoral students. Next one. And just to give you an overview, again, not much changes, except our diversity moves in the right direction. We have the same number of international students with the same country, although you might see in the ARCS Foundation that we are switching now between India and China, and this trend will be continuing. And this is not just here at UCSC, it is across the country. Next one. And here I hand over to Don, who will talk about co-led by him and Dart Newman, an initiative, and I will pick up later here the Envisioning Graduate Education for the Future. Don.
Thanks, and I want to say it's a pleasure to be here today and see all these ARC scholars, and truly everything you guys have accomplished and the recognition that you've received, it's very well deserved. So I want to talk briefly about a major initiative that the Graduate Division has embarked upon over the past year or so, and it's called Implementation Task Force for Inclusive Excellence in Graduate Education. We abbreviate that, the ITF, just for various reasons. A major effort here, if you can go to the next slide, please. Our effort here is we've done a very deep dive to evaluate using the data resources on our campus to evaluate how we support our students and how that support, financial support and other kinds of support is associated with student success. And not surprising, we've learned a few very important things that we're now pushing forward to implement in terms of how we can enhance graduate student education success. The one thing we've learned is, of course, the level of student support that we are able to provide is key in allowing our students to succeed. And I want to put a plug that our campus is one of a few UC campuses that provide guaranteed academic year funding for all of our doctoral students and MFA students. One of very few campuses nationwide that's able to do that. And we've recognized through this analysis that it's not just the ability to guarantee funding for academic support over 5 years, it's the level of support that's important. And I'm proud to say that our graduate students, particularly our doctoral students, among the best supported students in the graduate students in the entire country. So the level of support, of course, is hugely important, and we recognize that, and we're doing more as we speak to enhance that level of support. But the other thing we know is very important that we integrate with the financial support is the level and the quality of mentoring that we're able to provide. This is primarily at the program level through faculty PIs, and also the level of professional development resources and training that we're able to provide. And this initiative that we're in the midst of implementing right now in the graduate division is linking the higher level of financial support, enhancing mentoring at all levels, at the program level, peer mentoring across the campus, and linking that also to as professional development so as to optimize student success. And we can go to the next slide. On the graduate division website, we have now sort of retooled some of our resources that we are pointing students and providing guidelines for how to enhance learning at the program level. And we're also have totally retooled the package of our professional development resources. And we've identified key areas of professional development that we're trying to point out to students that's important for them to gain and skill sets in order to launch them on a successful career path moving forward. But I think the next slide starts talking about the envisioning graduate education for the future. Peter, you want to speak to that?
Yes, absolutely. And quickly, because you have all that available on the website as well. So next slide. Together with students, we laid out our key recommendations. And don't go through all of them. And the ARCS members are invited really to, and you will receive the strategic plan. It is basically what Dawn said. We have to take better care of you from the beginning to the very end, including the professional development and including funding and including really also internships and everything what we really can do for you. And of course, and as you can see here, elevating and enhancing support for graduate students off and off campus. Please go to the next slide. Then you really see how we face it. And you know that already, you fellow graduate students, that the main goal, the really what we're all struggling with, and time to come is, of course, housing. And we are trying that, that's goal number 4. We're really trying to do that, but we cannot really do it from tomorrow. We have to do it over the next years, and that is what this in regard to our goals. Next one, the strategic plan. This is really important. This is new. Our ARCS folks haven't seen that yet. This is really a completely new way of looking at graduate education, student-centric, and from a 360, from, if you look at the left side, encouraging folks like you to come to us to do research, and then 360 until we hopefully get you the wonderful job. And you're thinking of us like the ARCS Foundation is thinking of us and helping and giving back. So next one, please. These are some plans, and I leave that with you, especially in regard to new programs which we're doing. As I talked about diversity, we are a member, and I'm leading the 21 graduate schools at this HSIU here, the number 4 point. So we are tapping into new talent for the first time. And of course, we are working, and hopefully some of you will be a part of this recognition. We are working on graduate student recognition. We want to have a distinguished graduate student award for teaching, for research, and also for outreach communities. So this is coming on, very soon, hopefully, this year, and some of you will compete for it, as well as we want you to help us identifying the super mentor. So this is coming, the faculty and credit student recognition is coming online very soon. The last one, I think, and then we are all done, or was that the last one? Almut, right? And but again, we are sending you, yes, was the last one. We're sending you this presentation as well as the strategic plan. We are really at a wonderful time. And ARCS is just like one sign, one continuation of the support we need to make you successful in your credit studies and do discovery, do innovation, and afterwards think back of us at your good time at UCSC and the ARCS Foundation. Thank you.
Thank you, Dean Veal. Can you all hear me? It looks like I'm frozen. Okay, very good. Thank you, Dean Veal, and also Associate Dean Smith. Boy, that was very exciting to hear the plans and the execution of those plans and the support for ARC scholars. And of course, that is very near and dear to our hearts here at ARCS. But the support that you're providing the scholars and the commitment to that 360 wraparound is very, very exciting. And your enthusiasm speaks volumes. I just want to take a minute. I'm Chris Brent. I'm one of the co-presidents for our ARCS chapter in Northern California, to really congratulate all of the scholars in this year's cohort. You clearly are at one of the best universities, as just described, and you are the best of the best, having been selected by UC Santa Cruz for your ARCS award. We're very proud of you. We congratulate you. We look forward to getting to know you a little bit today and then through the year. And we hope to stay engaged with you, not only during your ARCS year, but in all the years to come, because you've earned this, not only in your achievements, but because of the belief in you that you will make an impact and will make the world a better place. And we want to be part of that journey and be rooting for you in every way, all along the way. And so with that, I just want to welcome you, congratulate you, tell you that we are honored to be on your team. And I'm going to turn it to Heidi Hutchberger, who is our co-vice president for university relations. Heidi?
Hi, folks. I mean, I'm telling you, I just love that introduction. In fact, Almut, also, the slideshow was brilliant. It really was. You don't mind if I rat you guys out and suggest that to other people. It was wonderful. But we're so happy to be here. And I'm telling you, it's really an incredibly impressive group of people. And I thought maybe you'd like to know a little bit about ARCS. Some of you do and some of you don't. We are a national organization with 15 chapters. We were founded in 1958, and I don't know if any of you know the story of why, but I thought I would read a little bit of background to you. This is from the NASA History Division, and let me just read this. The history changed on October 4th, 1957, when the Soviet Union successfully launched Sputnik 1. The world's first artificial satellite was about the size of a beach ball, 22.8 inches in diameter, weighed only 183.9 pounds, and took about 98 minutes to orbit the Earth on its elliptical path. That launch ushered in new political, military, technological, and scientific developments. While the Sputnik launch was a single event, it marked the start of the space age and the US-USSR space race. Why am I bringing that up? It's because that's what motivated a group of women in Los Angeles to found ARCS. They were exceedingly worried about the Soviet Union outdoing the US in the space race. And I thought all of you would find that, since you're in STEM and medical research, interesting as a background. Well, as Chris was mentioning, the Northern California chapter is celebrating its 5th decade of being science advocates. And we've awarded more than $1 million to our 7 universities this year. And specifically, we've awarded $175,000 this year to you folks. And as you know, you are 16 of our elite 84 scholars. And so I wanted to welcome everybody and tell you just how exciting it is. And I tell you, I really enjoyed that overview, Peter. That was really interesting. Thank you so much. I went to UCLA, and it makes me want to go back for graduate work. That's very old for me, but it was absolutely stunning. I mean, but very motivating, truly. And if there's anything that ARCS can do to help you along in terms of, some of the personal development, I mean, you should reach out to us because we've got a lot of folks that, have done some interesting things in their professional life for whatever that might be helpful. So I am going to turn this back to Amit after mentioning the fact that we look forward to seeing all of you at our symposium on April 22nd, which Peter alluded to. And hopefully that will be an exciting event where we can see you face to face. With that, I'm going to hand it back to Amit. Thank you so much.
Thank you so much. And we are going to roll the next slideshow, please. And Amanda, you're going to start.
Thank you. Hello, everybody. I'm very grateful to be here. My name is Amanda Carbajal, and I am currently in the last year of my PhD in the molecular, cellular, and developmental biology track here at UC Santa Cruz. Previous to doing my PhD, I did a Master of Science through SFSU and UCSF in San Francisco, where I studied neuroactive drug discovery. And in my first 2 years of my PhD, I actually worked at NASA Ames because they have a joint support group lab through UCSC. And so I was able to do my work there. But after the pandemic started, I switched into my current lab, where I study clinical microbiology, and I actually study uropathogenic E. coli that causes urinary tract infections, and when left untreated, it can go into your bloodstream and cause sepsis, which is very, very, it's a very big world threat, according to the World Health Organization. So I used a mixed methods approach. So I do whole genome sequencing, where I work with real patient samples to look at the mutations that exist in the uropathogenic E. coli that are allowing them the capacity to override the mechanism of action of different antibiotics that are typically used to treat these microbes. And so collectively, all our data will hopefully help all of clinicians around the world to understand better how to use other types of drugs that are actually effective against these mutated E. coli. And not only that, but our collaborators are going to use our data to derive better antibiotics for the next generation of microbes that will arise. And thanks to you all here at the ARCS, I'm able to focus on completing the data acquisition that's required for my last year of my PhD. And as soon as I finish that in a couple of months, I'm going to begin to sit and write all the data I've collected to put forth my first author paper that sheds light on all of the different types of drugs that I've studied and all the different mutations that I derived from the patient samples. This would not have been possible without your support, and I'm very excited to share more with you and meet you all in April. Thank you, and next slide.
Hello, my name is Anne Belke. I'm a graduate student in the Ocean Sciences Department. And a little bit about me, I grew up in South Dakota, so far away from the ocean. But I got my undergraduate degree at the University of Minnesota in genetics and ecology. And while I was there, I studied abroad in New Zealand, which is when I first took some marine classes and started to get interested in the ocean. I worked a few years before going to graduate school. So right after I finished my undergrad, I worked in a lab where I was working on some work kind of similar to what I do now, which is molecular ecology. I was working with plants. And then I started working for the Forest Service as a fisheries field biologist. And I did that for 4 years. And that's where I got interested in salmon, which brings me to my work now. I'm doing molecular ecology and conservation genetics work with salmon and steelhead, trying to learn more about the genetic basis of their life history traits to better improve conservation and management strategies. And thanks to the ARCS scholarship, I'm going to be able to I'm going to use the funds directly towards my research and fund some whole genome sequencing to be able to look at a genome-wide association study to try and get at the genetic basis of two key life history traits that I've previously found to be heritable, which are the age that the salmon return and the time of their spawning. And yeah, with that information, we'll be able to learn a lot more about the species and better understand their ecology. So thank you.
Next slide, please.
Hi, everyone. I'm a fourth year here at Santa Cruz. I'm a Cotto Robles Fellow as well. I did my undergraduate education at Princeton, and then I have a master's degree from Cambridge in astrophysics as well. Here at Santa Cruz, I work on exoplanet theory. That's planets around other stars, and I make computer models and do math about them. I also a coach for the UC Santa Cruz rugby team. And so the work that I do here at Santa Cruz is making computer models, which are not very cool looking, but I have to the left a photo of my code there, and that's what it looks like to be an theoretical astrophysicist, is lots and lots of computer work. And so I make models of supersonic winds that fly off of exoplanets when they're highly irradiated by their stars. And so these winds can actually strip entire atmospheres off of planets, leaving them as bare rocky cores. And so we want to understand the effects of these processes on the evolution of exoplanets. And it's a very large open question in our field, is understanding this process. And so especially this is amazing timing for the ARC to be part of this community. I'm so excited to meet you all, and especially the inspiring folks who are part of ARCs permanently. And this is an amazing timing for us because my advisor came on some hard times during the pandemic. Her health is not amazing. And so this generous scholarship is going to allow me to really make up the lost time and the lost networking connection opportunities that we had during the pandemic because we didn't have the funding to do so. So this is amazing to have that financial stress relieved. I'm really grateful.
Hi everyone, my name is Elsie Carrillo, and I'm a fifth year ecology and evolutionary biology PhD candidate in the Meta Lab here at UC Santa Cruz. I'm interested in studying the diving physiology and behavior and foraging ecology of aquatic reptiles like snakes. And I'm particularly interested in using garter snakes as a model to study the evolution of semi-aquatic or amphibious behavior. We recently discovered the use of Nereal air bubbles in the wild and captive bred garter snakes with different lifestyles. And so we are using an oxygen sensor to examine how rebreathing sustains submergence. This is an exciting and novel behavior that gives insights into the mechanisms that facilitate a semi-aquatic existence, and a project that is now possible with the generous support of ARCS. In the future, I aspire to be a biology professor at a research institution. Before realizing I wanted to teach in higher education, I actually pursued my master's in education and secondary science teaching credential from Stanford University as part of the STEP program there. And so then I ended working as a 8th grade science teacher in San Jose, where I grew up for many years. And so as creative outlets, currently I enjoy drawing, like scientific figures, and playing flute in the University Jazz Ensemble here on campus to connect with my peers. And I'm grateful, super grateful to ARCS for supporting me in my journey and affirming me and that I am capable of becoming a professor so that I can continue to mentor all students, including first generation and underrepresented groups, in developing their scientific identities through lab and field research opportunities. So thank you so much.
Hi, everyone. My name is Jillian Dorn, and I am a graduate student at UC Santa Cruz pursuing my master's degree in science communication. And so I am mostly interested in exploring health and human behavior through my writing, but this is the first year of the program, and so I'm still figuring out exactly what this is going to look like for me. But for the most part, this funding has enabled me to pursue my goal of communicating complicated technical material to general audiences. And so I'm interested in moving on after this program to do that either as a journalist or a writer, a communicator for an institution. And yeah, this program has been wonderful so far in learning how to developing basic skills for communicating, and I assume that it will continue to be. So I'm very grateful for this funding and this opportunity to be here exploring what it is that I will be doing next. Thank you.
Hi, Hi, everyone. My name is Amanda, and I'm a sixth-year PhD student in the Earth and Planetary Science Department. I'm an eco-hydrologist, and I'm interested in how water is stored and released from hill slopes as stream flow or plant water use. I'm currently a UC President's Pre-Professorate Fellow. In this role, I work with the UC Teaching and Learning Center to build professional development material for my department. Recently, I led a workshop focused on identifying and implementing strategies to improve students' quantitative confidence and literacy within geoscience classrooms. My ARCS research aims to think about how vegetation water use and timing influence subsurface water storage depletion. I'm currently working in landscapes that have one hill slope has trees and another hill slope has grasses. And so I'm thinking about how these different vegetation influence subsurface water storage and groundwater depletion. The ARCS Fellowship has really transformed my last year of my PhD by alleviating financial stress related to summer funding and traveling to my field site to collect my last round of water samples. Now I can more efficiently finish my PhD and apply to faculty jobs. So thank you so much.
Great. Hello. So, hi, my name is Yazi. I go by Yazi, but my full name is Yasmin Al-Shinawi, but I go by Yazi. So I'm really happy to be here and meet everybody. So just a little bit about myself. I'm a fifth-year PhD student from the METOX department. So that's the Microbiology and Environmental Toxicology department. My path to grad school was a little bit unconventional. I got my undergraduate degree at UCSC for general biology and decided around my my fourth year, like my last year, that I wanted to enter the microbiology field. So I worked as a post-bac researcher for about a year in the Ottoman lab, and then joined the Ottoman lab as a master's student. And I guess right before I was about to wrap up my master's degree, Karen, the PI of my lab, generously offered me an opportunity to stay on as a PhD student, and here I am today. So the Ottoman lab, we specifically work with a gastric pathogen called Helicobacter pylori, and that infects a large number of the population, and it can establish lifelong infections that cause gastric illnesses, so ulcers, and even stomach cancers and whatnot. So the World Health Organization and the NIH have classified the Helicobacter pylori as a high research priority due to that. And my project, I specifically focus on a gene called RSFS. So RSFS regulates the is a ribosome silencing factor that regulates the ribosome in Helicobacter pylori. And I'm studying the role of that particular gene and how it contributes to Helicobacter pylori chronic infections. So I'm very grateful for the ARCS scholarship. I had a lot of personal and medical setbacks this year, and with the support of the ARCS scholarship, I was able to publish my first literature review. I'm in the final stages of resubmitting an F31 application and my first author research article, and I've also been able to attend conferences as well. I was able to go to a molecular biology conference in Wisconsin recently, and I'm going to another conference that specializes in Helicobacter pylori in Germany this December. So I'm very, very grateful for the support from this scholarship to help me meet my academic and research goals. So thank you.
Hi there, my name is Ryan. I'm a fifth-year PhD student from the Earth and Planetary Sciences department. I'm broadly interested in leveraging the potential of the ocean for carbon dioxide removal. And for context, carbon dioxide removal is the process of basically just removing carbon from the atmosphere to mitigate future warming. My primary focus lies in exploring ways that we could potentially modify the ocean chemistry to enhance the natural carbon uptake that's already occurring. Outside of academia, I've been providing some consulting services to carbon dioxide removal companies. And in my free time, I like to cook, exercise, and basically just spend time outside. Any activity outside I enjoy. So my ARC research specifically, I'm trying to answer 3 specific questions related to this. I want to know how effective are these carbon dioxide removal methods? How efficient are they? And most importantly, are they safe? We need to know this stuff before we start implementing any of this stuff out in the field. And to answer these questions, I'm using a model that's built for the California current system off the west coast of the United States. And finally, I just want to say I'm very grateful for this funding and thank you. It's allowed me to dedicate my full attention towards research without the distraction of teaching. And obviously, the climate crisis is an urgent problem at hand. And I think the more time that I can put towards my research helps us move towards solutions to this climate crisis more quickly, hopefully. So thanks again. And I'm looking forward to meeting you all in April.
Is Julia here? He may not be here, so I will just let you look at the slide. I don't think it's the same effect if I read the slide. I think it's so much more effectful if the scholars present their own work and talk about their research. But Julia is in the Department of Ecology and Evolutionary Biology, and this is her slide. And I guess we can move on to the next one.
Hi, my name is Zach. I'm excited to be here. I'm a 5th year PhD student in the Department of Statistics. Some fun facts: I enjoy hi-fi audio and hot sauce, and I have a square number of teeth, 25. So, I'll let you figure out how that happens. My research is in a way about earthquakes, but being a part of the Department of Statistics rather than geology, really my research is on methodologies, ways of analyzing these types of data in a way that extracts the most valuable information out of what's available. And so those processes that I study are called renewal processes. There's a whole bunch of stochastic modeling theory that goes behind them, and that's where I make my contribution. But it's an exciting way of contributing valuable frameworks to geological and other sciences that sort of match the data template that these methods focus on. I'm really grateful for the foundation and for the opportunity to be a scholar. I work part-time. It's expensive to live out here. I have a family, and it's, tough when you don't have anyone to split the rent with, right? So one of the primary ways that the fellowship has impacted me is by allowing me to spend less time working on that part-time job and being able to dedicate that time to finishing my PhD in a timely manner, as well as just spending time with my spouse and kids. So I'm very grateful for that, and thank you.
Hi, everyone. I'm Alex Kramer. I'm a grad student in biomolecular engineering. And first of all, I just want to say a huge thank you to the ARCS Foundation for their support through this scholarship. I'm currently in the 4th year of my PhD, working in Russ Corbett Dedek's lab, and my research is centered on pathogen genomics and building evolutionary trees of pathogens at the scale of millions of genomes. So a lot of this research was first started due to the widespread global sequencing effort during the COVID-19 pandemic. And we've now sequenced over 16 million genomes, complete SARS-CoV-2 genomes. So this huge amount of data really pushed the boundaries of what existing bioinformatics platforms could handle. So to respond to this, our lab developed a new way of inferring phylogenetic trees from these huge amounts of data. And we currently host a daily updated global tree for SARS-CoV-2 that, as far as I'm aware of, is the largest tree ever constructed from genomic data. So what I work on now is developing algorithms and web applications for fast analysis of data sets like the one for SARS-CoV-2 that contain potentially millions of pathogen genomes. And these tools are used by public health officials to help track and mitigate the spread of pathogens. And in the future, I'll be working to hopefully streamline this process by making tools faster and more accurate and hopefully more accessible to researchers around the world. And beyond just helping support my research, the ARCS Award will be very helpful in helping me find more time to dedicate to mentorship, science communication, and also building connections within the scientific, and public health community. So I'd like to say thank you again to the ARCS Foundation for this opportunity, and looking forward to a productive year to come. Thanks.
Hi, everybody. My name is Suzanne Lipton. I am a 5th year PhD candidate in the Environmental Studies Department at UCSC. And my broad research interests are in agroecology, biodiversity, climate change, and soil microbial ecology. Prior to starting my PhD, I worked at several nonprofits in the realm of environment and agriculture, and worked for several years as the assistant director at the Earth Institute Center for Environmental Sustainability at Columbia University. My interest in agroecology actually stems from an earlier career as a pastry chef in farm-to-table restaurants, where I became aware of the intricacies of sourcing sustainable food and started to wonder what the environmental benefits were of items such as free-range chickens, grass-fed beef, or integrated pest management peaches. This ARCS funding will help me complete my dissertation research. Big picture, my research examines above and below ground interactions in agroecosystems. And in particular, I'm interested in how above ground management can impact biodiversity, climate change, and soil health. And more specifically, my dissertation project investigates how dung beetles, which are present in California, not many people know that, affect the soil microbial community and associated nutrient cycling in pasture lands of the Central Coast. And I'm also looking at how management practices, so management of the cows and surrounding and the pasture lands, as well as the surrounding landscape, affect dung beetle populations. And ARCS funding is going to give me much needed time to focus on my research. So as we all know, half of the time of a PhD student is usually spent working either as a TA or a GSR, and ARCS funding will give me time where I can just do my research, which is one of the best gifts I could possibly think of while getting a during my PhD. And I will use this time to complete data analysis and write manuscripts. So I'm really thankful for this funding and really appreciate the support while I complete my research.
Hi, everyone. My name is Madeline Reinsel, and I'm a graduate student in the Science Communication program here at UC Santa Cruz. I'm originally from Virginia and have a bachelor's degree in environmental science from the University of Virginia. So before coming to Santa Cruz this fall, I worked in an environmental science lab at the College of William & Mary for several years, where I was working on research and communication projects. So like Jillian, who's also in my class, I've only been here for a short time, but I've already been able to work on several cool stories around Santa Cruz, including a feature in the Santa Cruz Sentinel on the damage to Westcliff Drive last winter, and a story on endangered amphibian relocations. Some of my hobbies outside of work and school include sewing. I make a lot of my own clothes, and doing any number of outdoor activities. I'm just overall very grateful for the support of the ARCS Fellowship, which is funding a large portion of my tuition in this one-year program. That's it for me. Thanks so much.
Hi, everyone. My name is Dominic Sanchez. I'm a quite informal guy, so you can call me Dom. I'm a graduate student here at the Astronomy and Astrophysics Department. My research focuses on developing astronomical instrumentation for exoplanet science. And it's really an exciting time for astronomy because we're beginning to develop and deploy instruments that may be able to answer interesting fundamental questions like, are we alone? Are there other planets that may be harboring life? Are there more habitable planets? And so the next generation of telescopes will be able to detect more planets outside of our solar system and characterize them. But to fully exploit telescope performance, we need to further develop the technology to compensate for atmospheric turbulence. And so as light from these planetary systems propagate through our atmosphere, they become distorted and blur the images of these systems. And therefore, we're not able to distinguish that planet from its host star, and therefore decreasing the resolution of our telescopes. So in a nutshell, my interest is really developing instruments that compensate for these optical aberrations. And this set of technologies called adaptive optics, or AO. And so been pretty excited because we recently developed and deployed a novel sensor on Chain Telescope at Lick Observatory here in Mount Hamilton, California, and we've confirmed its feasibility. But in all, adaptive optics will be really integral part of future observatories. And I'd like to thank the ARCS Foundation for funding my work and to continue the hunt for habitable exoplanets. But outside of my work, I really enjoy fishing, camping, and I recently picked up archery. So I try to stay outdoors too, since we can be a little bit of lab rats. Thank you. Next slide.
Hello, everyone. This is Mace Mohammed Salih. I am a final year PhD candidate at the Department of Molecular and Cell Developmental Biology. I am a California Institute of Regenerative Medicine fellow. I am also a former trainee at the Tobacco-Related Disease Research Program, which I am generous for these contributions that support my research. I am also very passionate about mentorship and creating community within my graduate program. Fun fact about me is that I am a mother to 2 children, 8 and 2 years old. So whenever I'm not in the lab pipetting, that's where I spend most of my time. However, in the lab, I focus on immunology, specifically how our bodies respond to pathogenic infections. So whenever we are attacked by a pathogen, there is a fine-tuning of our response that needs to happen to make sure that there is not too much of a response. If there is too little of a response, we all know the pathogen will take over. However, if there is too much of a response, this could also be bad for us because it could drive chronic autoimmune diseases. These are things like rheumatoid arthritis, systemic lupus erythematosus. So our bodies could quite literally overactivate the immune response and result in really bad consequences for us. And in the lab, we study how this tight regulation happens. An example is an RNA splicing protein that I have found to promote a very major inflammatory signaling cascade in our cells. This protein has also been found to correlate with rheumatoid arthritis. So we obtained samples from rheumatoid arthritis patients, and we've seen that this protein actually drives and promotes a lot of the immune overactivation in these patients. So we are working on developing this protein as a therapeutic target. And in the future, this could hopefully alleviate a lot of the patient's pain and suffering and help improve their lifestyle. So all of this research is happening in the lab, and which all wouldn't have been happening without the generous contribution of the ARCS Foundation. I use the generous grant from ARCS Foundation to support my family, to provide childcare for my children while I'm in the lab conducting my research. So I'd like to thank you all for your generous contribution, and I will meet you all soon in April. Thank you. Next slide.
Hi everyone, I'm Navid. I am a PhD student in Electrical and Computer Engineering department. I was a former chip designer here in Silicon Valley at Advanced Micro Device and Sound Microsystems, working on next generation computing chips. I started my own company, which is called Ectosonic, which is the focus of my research here in the ECE department. And the idea is, it's a new trend in medicine called focused ultrasound or curing with sounds. They send these harmless sound waves into the body for treating numerous medical conditions non-invasively without any surgery or chemotherapy in case of cancer. There is over 170 medical conditions that are being treated with this revolutionary technology. This number is actually 4 times increased in the past 4 years. When I actually started Ectosonic, it was, I think, less than 50 conditions. Now it's 170 conditions. And I'm very grateful for this award, which helps for my cause here and continuation of the work. And basically, all I can say is this is the future of medicine, and we have a dramatically cost-effective and superior performance technology with the driving electronics for these transducers that generate acoustic waves that basically focus the energy into the body, and it has other non-medical applications. We have launch part of the technology as of now, and we are working towards launch of the phase array, which is the major technology that we have. Thank you for that.
And Diana, can I please ask you to go back to Julia's slide? Everybody, the students really are juggling teaching obligations, lab assignments, and a million other things. And Julia was only able to join us now, but she's here, and I really appreciate that she's going to be able to present her slide.
My apologies for joining late. It was a tough morning, family emergency. But I'm very excited to be here. My name is Julia. I'm in my last year of my PhD in Dr. Kathleen Kay's Plant Evolution Lab in the Ecology and Evolutionary Biology Department. And I'm really passionate about understanding the role of hybridization in plant evolution. There's increasing evidence that hybridization is actually common across the tree of life, despite previous assumptions that it was very rare. And so I'm very curious about how this affects how species diverge and change across the environment. My most recent award was for Best Student Presentation at the American Society of Naturalists Annual Conference. I also just received a Chancellor's Fellowship, I believe, which is a dissertation quarter fellowship. I love botany hikes. I'm a big time plant nerd. I love snowboarding and surfing. Yeah, those are my main hobbies. I'm also terrible at watercolor, but really enjoying it. My ARCS funded research has helped us understand how the tropics hold such immense plant diversity. And this is because I study hybridization through both the ecological lens and genetic lens in two closely related tropical spiral ginger species. And my research goes to understand how they're able to be maintained as distinct species despite their close relation and the hybridization that does occur between them. And this is something that occurs broadly across tropical plants that are so diverse. Close relatives often live in close proximity and can hybridize, and yet don't do so much that we lose the diversity that exists in the tropics. So it's wonderful work. I get to travel in Panama and Costa Rica, and it's been extremely rewarding. The ARC Scholarship has released me from teaching in my final quarter, next quarter, which has been an incredible gift and given me the opportunity to spend time developing postdoctoral fellowship applications. I've applied to several at Stanford and several at UC Davis with some folks whose work I've idolized for years, and I'm very excited about those opportunities. And the honor of this award, the ARCS Award, and the prestige it holds will certainly also help, hopefully, in getting one of those postdoctoral fellowships. And then, of course, now that those are mostly off of my plate, I can turn to actually publishing my final chapter. I've published my previous chapters, and that's the last one that's waiting to be seen by the world, and also the one I care about most. So getting to spend the time to focus truly on writing without splitting my time between teaching has been a real gift, and I'm extremely grateful. So thank you all. And thank you for letting me tag on at the end here today. I appreciate it.
Thank you. And that concludes the slides and the sharing. Thanks to everybody for sharing their stories, their research, and for participating. And we open it now to questions. I would suggest, because we don't have that much time, that you just unmute yourselves and ask questions to a scholar, to the scholars in general, or whatever you're curious about.
I have a question concerning hybridizing. There's been this concern about gene altering, but hybridizing has a very different kind of is a different world, right? Can you explain that to us?
Will you explain what you mean by gene altering specifically?
Oh, nobody wanted tomatoes with their genes altered, right? Remember that big fuss?
Sure.
But the plant was changed. So how does that differ from hybridizing as you're looking at it?
Great question. So when you think about altering the specific genes in, say, a crop, that is going in and editing the sequence of letters that make up our genome in a very specific way. So there's a gene, we know it does something, and they go in and they change a few base pairs, change some of the language basically in your genome to change a little bit what the product is and what the plant ultimately looks like. Hybridization is sort of a wholesale reshuffling of the whole genome. So you have maybe 2 different kinds of tomato and you cross them, and now the new thing that you get is a mix of everything that was in one and the other. And what I'm studying is basically what happens after that occurs. And in the wild, it can be that those new combinations are really beneficial and they outcompete the 2 original parents, or it can be that that's broken up some really different adaptations in the 2 species that enable them to live where they do, and has created something that's not viable and can't live in the environment because it's sort of not well adapted to either situation. And so it's this interplay of how the genome reshuffles and then how the environment acts upon that reshuffling in what I study. In breeding in crops, they do a lot of hybridization and natural or artificial selection, which is basically the same thing I described, only instead of the environment saying, yes, you get to make it, or no, you're not fit, it's humans saying, yes, you're delicious, we'll keep you, versus no, you're, puny tomato that we don't want.
Thank you. Thank you.
You're welcome. Great question.
Alex, this is Heidi Hutchberger. I'm curious about your sequencing and what has it taken in terms of advancements in software and hardware to allow you to make those kinds of gains? I mean, it's got to be a combination not only of just intellectual brilliance, but also technology.
Yeah, so the sequencing was really just like a global widespread effort that generated a huge data set that computationally, which is what I work on, is really hard to deal with. So before the pandemic, phylogenetic trees, evolutionary trees, which are what we work on building, were really very small compared to the scale of 16 million sequences that we have now. So a lot of the existing methods just would take years to run, so just not really feasible in a pandemic setting. So the big innovation of how we construct trees is an algorithmic technique that instead of essentially building the tree from scratch every time you get new data, every day in the pandemic, you add the new samples that are sequenced and collected onto an existing tree that grows over time, which takes far less computational time in the long run over time. And also for, at least for SARS-CoV-2, has been shown to be accurate enough that it's comparable to the previous approach of sort of building things from scratch all the time. So that's the main advancement, I would say.
That's the secret sauce.
Yeah.
Thank you.
I am afraid we are at time, so there was just not enough time to do Q&A today, and I feel very sorry for that. But I do want to allow the ARCS Foundation to say some closing remarks. So I'll hand it over to Susan.
You're muted.
Hi, I'm representing the Scholar Relations Committee of ARCS. It is so delightful to meet all of you and to hear about the UC Santa Cruz program, which is so exciting in its innovation and its development. All of your topics are just fascinating. You are our stars. There are several other committee members on today, also Jerry Del Vecchio, Julie Reese, and Carmi Ticehurst. We all hope for your success. It is our total focus as Scholar Relations Committee. You are part of a community in Northern California that has reached 11,000 students at this point. So over the years, we hope to facilitate your connections with each other. And I know one of the benefits of this meet and greet is that at least you get to see each other. I'm not sure whether on campus you bump into each other or not, but there are 7 universities among your cohort. We get you all together April 22nd this year. at the City Club in San Francisco. So look forward to that. We will prepare videos of your work. You will have your posters and you will get to see each other in a hopefully very party, fun, productive, rich environment. We look forward to other ways of supporting you. You might check out the LinkedIn connections that ARCS has. Bravo. We all benefit from your research. Thank you. Thank you.
That's great. And I'm Judy, and just as Judy Webster, and as a final sign off, my thanks to the university, to the administration, to the scholars, and to all of the ARCS members which make this possible, and do keep in touch. Many thanks.